Hello, and thank you to everyone around the world for joining us for this week's lab meeting. My name is Kristen Aboud. I am the science editor at the Human Vaccines Project, and I will be your moderator today. Last week, the US Centers for Disease Control and Prevention relaxed their COVID-19 guidance, now stating that fully vaccinated individuals do not need to wear masks indoors or outdoors in many situations, marking a major shift in the country's progress in fighting the pandemic. Though for many, this seems like a step toward normalcy, the CDC is still urging caution for those individuals who remain unvaccinated, including children under 12 and those who are immunocompromised. Other countries around the world with high vaccination rates are also starting to see declines in COVID infections and are similarly loosening restrictions. However, in many parts of the world, vaccine access remains severely limited, and globally, infection rates are still on the rise. This glaring disparity has shown just how critical vaccines and access to these vaccines is in an effort to control the pandemic globally. The US government announced plans to send 80 million excess vaccine doses to help beleaguered countries, but many say this isn't nearly enough supply to stop the virus from wreaking havoc in much of the world. One vaccine the US will be sending abroad is the mRNA vaccine developed by Moderna in partnership with scientists at the Vaccine Research Center, which we will hear more about in just a few moments. In the meantime, many companies are also developing second-generation COVID vaccines that have favorable profiles for global distribution. Before we get started with today's presentation, I wanted to note that the application portal is still open for the Michelson Prizes, Next Generation Grants. Presented by the Human Vaccines Project and the Michelson Medical Research Foundation, the Michelson Prizes are given to support promising young researchers who are applying disruptive concepts and innovative methodologies to advance human immunology, vaccine discovery, and immunotherapy research for major global diseases. We're posting the link to the Michelson Prizes in the chat, so please visit the site for more information or to apply. And just one more note before we begin. The information presented today includes some pre-published data that is currently under peer review. At the request of our speaker, we ask that you not reproduce or disseminate the data presented. This session will be recorded and made available on our website and our social media channels for you to review. With that, I am happy to introduce today's speaker, Dr. Robert Seder. Dr. Seder came to the US National Institutes of Health in 1989 and performed his postdoctoral training in the Laboratory of Immunology at the National Institute for Allergy and Infectious Diseases under Dr. William Paul. Dr. Seder was subsequently appointed as chief of the Cellular Immunology Section at the Vaccine Research Center in 2000. Dr. Seder has focused on the cellular and molecular mechanisms by which vaccines and adjuvants mediate protective immunity in mouse and non-human primate models of HIV, malaria, tuberculosis, and cancer. His focus has been on T-cell quality and how the route of vaccination can be used to generate protective T-cell immunity. Recently, Dr. Seder has also focused his efforts on the discovery of monoclonal antibodies to prevent malaria infection. He led the first in-human clinical studies of intravenous vaccination with an attenuated malaria vaccine and of a monoclonal antibody to prevent malaria infection. Over the past year, Dr. Seder has been intimately involved in COVID vaccine development with the Moderna mRNA vaccine in the non-human primate model of SARS-CoV-2 infection. This work has focused on developing immune correlates and mechanisms of protection, understanding the durability of protection, and determining how well the vaccine will protect against viral variants. During the presentation, please send me your questions using the Q&A function in Zoom. I will ask our speaker a broad selection of your questions after the presentation, and we will have about 25 minutes for discussion. It is my pleasure to now introduce Dr. Bob Seder.
Thank you very much, Kristen. And it's a great pleasure to be presenting for all of you. And so let me go to the talk. Okay, I should first mention that I'm not going to talk about correlates. I'm just going to focus on a study with the variants. We can talk about correlates later, and I have some slides on correlates, but I think it's just best to focus on the variants because I think that's the greatest concern right now. And so the focus of the presentation will be to review some data that we generated a year ago with the Moderna mRNA-1273 vaccine in non-human primates against the Washington isolate, and then get into some background of variants of concern with a specific focus that at this stage, B.1.351 is a priority. Of course, most of you are aware that we're now shifting after emphasizing B.1.351, we're now shifting some of our emphasis toward the Indian variants, and we can talk about that at the end. And then I'm going to present in detail a mRNA-1273 vaccine study in non-human primates against the B.1.351 variant. So just as a background, the mRNA-1273 vaccine was designed by Barney Graham with help from Kazmiki Corbett and Jason McClellan. Roughly January 13th of 2020 was sent to Moderna, and they made the RNA vaccine encoding a stabilized spike called S2P. And the S2P stabilized spike is also encoded in the J&J vaccine, the BioNTech Pfizer mRNA vaccine, the Novavax vaccine. The only vaccine of the Operation Warp Speed 6 that does not contain this is the AstraZeneca. But it's full-length spike that's stabilized. It's encapsulated into a lipid particle for stabilization. So that is the vaccine. In our first study that we did in May of 2020, we gave monkeys 10 or 100 micrograms, which is the approved human clinical dose, twice at 0 and 4 weeks. And then we challenged them 8 weeks later with around 8 times 10 to the 5th PFUs of the Washington strain given intranasally and intratracheally. And then we follow them at days 1, 2, 4, and 7, or 2, 4, and 7, and taking nasal swabs and BAL for subgenomic RNA is one readout for protection. Animals are then euthanized 7 or 14 days later, and we look for inflammation in the lungs as well as detection of viral antigen. And so this is just a summary of the protection data. And what you'll notice on the left is this is the subgenomic RNA values in the BAL. And you can see in the control animals at day 2 that all of them have around 10 to the 5th. But in both the 10 and 100 microgram dose groups, even by day 2, 7 of 8 animals had undetectable lower airway subgenomic RNA. So there's really, this gave us great comfort at that time that the RNA vaccine in this model was completely protective in the lower airway. We were also delighted when we looked in the nasal swabs that 2 days after a challenge in the 100 microgram dose group, there was also undetectable subgenomic RNA, suggesting that at that dose, not only were we getting lower airway protection, but upper airway protection. You can see that 5 of the 8 animals had detectable subgenomic RNA at the 10 microgram dose group at day 2, and then by day 4, 6 of the 8 had controlled. So to summarize that, we said that this showed that the ability to limit viral replication, both the lower and upper airways, has important vaccine prevention of both disease in the lower airway and transmission. And I think this study was the first to suggest to us that protection in the lower airway may be achieved easier than the upper airway and may be dependent on the dose and immunogenicity of the vaccine. So this, of course, is the Kaplan-Meier for the efficacy and safety of the mRNA in humans. And you can see that you had 94% protection against mild infection and a complete protection against severe infection. So indeed, the primate model was modeling what we achieved in humans with no severe infection and very high level protection, even against mild infection. So now I borrowed this slide. This was made by my colleague, Nicole Doria Rose, is what about the variants? So the questions are they more contagious? Are they more deadly? Do they cause more reinfections? And most importantly, will vaccines still work? So the answer to those are yes, B.1.1.7 is more contagious. Are they more deadly? Well, there's some data that perhaps it's more pathogenic. Do they cause more reinfections? Well, perhaps. It appeared that people in South Africa were infected after they had been infected with an earlier strain. And will vaccines still work? And the answer to that is yes, but they might be reduced, and it depends on whether it's mild infection or severe infection. So this is just a summary of what we consider to be some of the major variants. I haven't included the B.1.617 from India, but obviously that's going to be in the next, in the next couple of weeks, we'll have a lot of data on that. But what you'll notice is we focused on the South African variant based on the fact that they have these multiple mutations in the RBD. 484 is notable to be resistant to certain monoclonals and is primarily responsible for that virus being less neutralization sensitive. And 501 is important for spread. And then you see all these other mutations or lesions in the N-terminal domain. So in our mind, in terms of prioritizing the South African variant and to some extent the Brazilian variant up until a week ago were the primary variants of concern and ranked highest. And I should just point out, since this is the global call, that the development and understanding of the South African variant came from brilliant work from Alex Siegel, Tulia Oliveira and Penny Moore in South Africa, who developed a system of really tracking these things. And so it really is kind of a global effort. And I've listed a bunch of references of those people who have done work on showing that either convalescent serum or vaccine serum shows a marked reduction in neutralization against the South African variant. And this is shown here in a publication from Kai Wu from Moderna, where you can see if you compare neutralization profiles. So this is mRNA-1273 vaccinated individuals serum roughly after the second dose, and you compare it against the benchmark D614G, and you can see that there's very little change in B.1.1.7. But when you look at P1, the Brazilian variant, or 1351, you can see a 5 to 10-fold reduction. So this clearly tells you that indeed vaccine serum is less neutralizing against these variants. It's 5 to 10-fold. But I would note that the neutralizing titers are still 1 to 300. And so a term that John Moore coined called spare capacity highlights the fact that we may be well above a protective threshold for protection. So even though you read that things are 5 to 10-fold reduced, they're still relatively robust, and I'm going to show you are able to confer protection. And so this is just a kind of a cartoon to highlight how we think about sensitivity to serum with the variants, and this is why we focus on the RSA variant. So the goal of the studies today will be to develop the NHP model for B.1.351 infection, which hadn't been done before, and then assess immunogenicity and protection of the current vaccine 1273 against the variant. And the focus will be of how the dose influences immunity protection. So we're going to use 2 different doses. And importantly, we're going to compare a single dose compared to the 2-dose regimen. And for most of you on the phone, you can understand that there's been a lot of discussion about whether you apply single-dose vaccines rather broadly and then bring people back when the second dose is available. So I think having data on comparing 1 versus 2 doses is important, especially in relation to the variants. So here's the study. So as before, we give a primate a boost at 0 and 4 weeks apart. We give 100 micrograms twice, which is the approved dose for under emergency use authorization, 30 microgram a dose, which the immune responses at 30 micrograms in monkeys really models what is seen with 100 micrograms in humans. And then we gave 30 micrograms once. And then we have a group of naive animals. There's 8 per group. And so either 8 weeks after the prime boost or 12 weeks after the single immunization, animals were challenged with 5 times 10 to the 5th of a B.1.351 variant by intranasal and intratracheal. And then we look, as I said before, in nasal swabs and bowels at day 2, 4, and 7 for viral replication. We take the animals down at day 8 and 14 for pathology. And across here, we do sampling to measure immune responses. So I'll show you data from blood, bowel, and nasal washes after vaccination and prior to challenge. And then I'll show you some post-challenge data in nasal washes and bowel for antibodies. So the first thing is we did a temporal analysis of the effect of the prime and boost on pseudo-neutralization against 1351. So you can see, and this was done by the team at the VRC and also by our colleagues at Moderna, that against D614G, with a single immunization of 30 or 100 micrograms, you readily get neutralization against D614G. And you can see this rather dramatic boost in those animals that got the second immunization, such that at the time of challenge, it's about 1 to 5,000, quite dramatic. You can see the single immunization actually does fine over 12 weeks and is close to about 1 to 500. When you look at B.1.351, it's quite different. You detect no detectable pseudo-neutralization against 1351 after the single shot, but after 2 shots, you get quite robust 1351 titers at the 130 microgram dose group. And you'll notice here in the single dose group, there is no detectable pseudoneutes at the time we will challenge. So 2 doses are required to detect pseudoneut against 1351. So now we developed, or MSD developed, a 10-plex assay where we can measure binding titers to the Washington spike or RBD, or the 1351 spike or RBD. And this is done by Adrian McDermott's group at the NIH. And you can see for the Washington, you have very nice binding titers at 8 weeks after the prime booster, 12 weeks after the single, in a dose-responsive manner. And again, the single dose group has less binding against both spike and RBD. You'll notice that they also induce very good binding titers to the 1351 spike and RBD. I haven't plotted them on the same curve, but there is a modest decrease in binding to the spike and RBD compared to the Washington strain. We also extended this, since this was a 10-plex, and included binding to B.1.1.7 and the P1 variant from Brazil. And it's basically the same thing I showed you. You have very good binding that's dose responsive between 100 and 30, and then the single dose binding titers are less. Also, you'll see that the binding titers to P1 do appear to be a bit less in the RBD than they are to Washington. We then look at the time of challenge. In addition to pseudoneutes, we also did viral neutralization and compared them to D614G. So you can see at the time of challenge, which was 8 weeks after the prime booster, 12 weeks after the single, while I showed you that the single 30-dose group gave you detectable titers against D614G in the circles in blue, there was nothing detectable against 1351. By contrast, both at the 130 microgram dose group, there's robust D614 titers, and they again go down 5 to 10-fold, but again, they're close to 1,000, between 500 to 1,000 to each of these 2 dose groups. And it's the same thing whether we use a lentivirus that is done at the VRC, a VSV pseudovirus, which is done at Moderna, or this live virus neutralization assay done by Mehul Sutar at Emory. So again, in the live virus neuts, there's no live viral neutralizing titers with the single dose, but there's very robust, or between 200 and 300 with the 30 and the 100 microgram dose group times 2. And this is all done at the time of challenge. So this just summarizes that there is a 5-fold reduction in neutralization of 1351 versus D614G. Neuts are still 1 to 300 against the 1351, and there's no neuts with the single dose. So now let's look in the site of action. So we measure IgG and IgA in the bowel and the nasal swabs after immunization and prior to challenge. You can see in terms of binding to the Washington or the 1351 spike, there's very good detection of IgG in the bowel, both at the 130 microgram dose group times 2, but there's much less with the single dose group in the bowel. When you look at IgA, it's the same trend, although the IgA responses are lower than the IgG responses. When you look at nasal wash, we see the same dose responsive trend with 100 microgram being better than 30 times 2 and much lower with the single dose. So we have IgG in the knobs, we have IgG in the bowel. And when we look in the nasal wash, again, It's much lower than IgG. And the only thing that looks like there's a trend toward being higher than the controls is in fact the 100 microgram dose group times 2. Finally, we do, of course, measure T cell responses. This vaccine, which really, this goes back to how Drew Weissman and his colleagues, Norbert Party, designed the vaccine, is in these nucleoside modified RNA. Drew was smart enough to realize that by altering the sequence, you could not only get Th1 responses and CD8s, but you could get persistent antigen, which would lead to, or more persistent antigen, which would lead to TFH responses and IL-21, which would obviously be good for antibodies. We've published this before, and so have many other people, but you can see that there's a dose response for Th1 with 100 being better than 30. There's no Th2s, but there are these nice TFH responses, both at the 130 microgram dose group, as well as IL-21. So these are very good responses, not only potentially for mediating a biologic effect against the virus, but for optimizing antibodies. We did detect CD8 responses, but only in the 100 microgram times 2 dose group. This is the 12 circulated antibody response, 1351, 117, and the P1 strains. Newts against 1351 required 2 doses. The T cell responses induced Th1 and TFH in a dose-dependent manner, and CD8s were only induced with a 100 microgram dose group. So now let's go to the challenge. So to do the challenge, we had to get an isolate and characterize it for challenge. So Andy Pekosh at Hopkins had isolated a strain from a subject that he had sequenced and shown that it was similar to the 1351 that was based on the South African strain. And he expanded it in viro cells, so there was a single passage. And then he sent that to Mehul Sutar Emery, who expanded a second time. So we're using the second passage, and we're calling that JHU P2. It was sent to the Vaccine Research Center, where Eli Boritz and Danny Duick sequenced it to assure that it contained all the necessary mutations compared to the benchmark 1.351 strain. We then did a dose titration in hamsters to show that the virus was pathogenic. And then we did a dose titration in monkeys to show that, again, we could achieve subgenomic RNA viral replication in bowel and nasal swabs. And then we chose a challenge dose for the vaccine study. So this just summarizes what I just showed you. Here's the 1351 Pango reference with the notable mutations in the RBD as well as the NTD. And here's the challenge stock that we used that was sequenced by Danny and Eli. So you can see it's a, quote, pedigreed virus. Here's the hamster weight loss data. So you can see in a dose-responsive manner that the hamsters all lost 15% at 10 to the 3rd, 10 to the 4th, and 10 to the 5th. And then when we gave it to monkeys, you look in the bowel and measure sgRNA for E, or the nasal swabs, you can see it 2.4 times 10 to the 5th, or 2.4 times 10 to the 6th. There's about 10 to the 5th to 10 to the 6th logs at day 2, and they remain relatively stable through day 6, both in the bowel and the nasal swabs. Our prior analysis with the Washington strain would show that the control in naive animals often by day 6 was complete. So this suggested that perhaps at these doses with this virus, there was more persistence of the South African strain. But this together showed that the strain was pathogenic. And based on this, we chose a concentration of 5 times 10 to the 5th to be used. And I think that I'm going to bring this up at the end, but I think this is a very conservative approach. I think we're probably giving more virus to induce higher subgenomic RNA that might be naturally transmitted in people. So here's the money slide. When you look following challenge with the 1351 strain at day 2, you can say 6 of the 8 animals that received 2 doses of 100 micrograms have undetectable subgenomic RNA. So that's fantastic. And these other 2 have really, it looks like 4 logs reduction. So the standard dose of the vaccine that's currently approved gives very high level protection in the lower airway against 1351. You can see at 30 microgram times 2, it's still statistically significant. It's probably four to 5 logs, although these two animals didn't do quite as well. You can even see that in the single dose group, there is a reduction in the bowel. By day 4, all these groups are statistically significant compared to the control groups. And by day 7, the majority have no detectable RNA. And I can tell you that you can see again by day 7, there's still 5 logs in the control animals, which again suggests that either based on the dose and or this virus, there's a bit more persistence than we had seen with Washington. So this is good news that lower airway protection with our current vaccine will be achieved against this variant. When you look in the nasal swabs in the subgenomic RNA, it's a different story. And I alluded to this in my first slide, that you can only see the high dose group gives about 2 logs at day 2. And it really takes till day 7 till you'll see that all the vaccine groups are able to control in the upper airway. So clearly the upper airway is less than the lower airway, as I had alluded to before, and I think is consistent with a lot of the vaccines studies that have been done in South Africa with the J&J and the Novavax, where they see less protection against mild infection than they had seen in other areas, but still, in terms of the J&J, had very good protection against severe disease. So again, it tells you that the threshold for upper airway protection transmission might be different, especially with these variants. We then looked at Biocall with Hannah Anderson and Laurent and Mark Lewis were able to quantitate the virus from the bowel. Since the sgRNA is a very sensitive measure, but this gives you a biologic measure of whether you can grow virus out. And again, you can see in these two groups, there's no virus you can grow out at day 2. And in many of these animals, the viral outgrowth is very low. So this tells us that the subgenomic RNA data in this data, which I can show you are highly correlated, and that the bowel titers are correlated, and there's no virus culturable with subgenomic RNA less than 10 to the 4th. So you have a measure of 10 to the 4th, but at least by this value, there's no culturable virus, which tells us that, and I'm going to show you the pathology in a minute, that these are highly protected. And so this is the pathology. And so you can see when you look at H&E staining, or this was done by Ian Moore, where he measures detection of SARS-CoV-2 antigen, you can't see any inflammation or antigen at the 130 microgram dose groups given twice. There is more inflammation with 30 micrograms times 1, and we do detect viral antigen. So this is consistent with the subgenomic RNA data, and the viral load data, that in fact, these are highly protective in the lower airway. And these are also protective, but there is virus down there and a little bit of inflammation. And you can see there's much more virus in the control and much more disordering and inflammation in the control. So finally, let me close with some kinetics of antibody responses in bowel after vaccination and challenge, because I think it gives us some insights into mechanisms of protection. So you can see that the red arrows show the pre-challenge responses, which I had shown you before. So you can see that the 30 microgram dose group, there's no IgG or IgA, or very little detected. But you can see at the 30 and 100 microgram dose group, there's really a lot of IgG, or much higher IgG detected in the bowel. And you can see post-challenge at day 2, 7, and 14, there's no change at all. So these animals in the bowel are completely protected, and there's no increase in bowel IgG responses. By contrast, you can see that in this dose group where I showed you there was some virus, by day 7, there's an increase in IgG, but especially at day 14. So there's an anamnestic response that occurs that's starting at day 7 to day 14 and consistent with them seeing virus. I also showed you that there was some protection in these animals. So it's interesting that you might have some protection in animals in the lower airway, but virus gets through and actually you get these anamnestic responses. And you can see these responses are actually higher than these other dose groups. And this is a point we recently made in a correlates paper, that it's possible that you might have some protection with vaccines that aren't as robust, and then the virus itself will give you a boost. And I would say that we haven't done it in this study, but when we've taken groups of animals like this and rechallenged them, they're completely protected in the upper and lower airways, suggesting that in some cases, the infection might boost immunity. When you look at the, I'm sorry, this is probably a copy of the slide, but it makes the point that I just made that pre-challenge, there's nothing here. And these are much higher, and then post-challenge, these are elevated. So this just summarizes what I just showed you, that pre-challenge, these are the highest titers and they're not changed. And post-challenge, there is an anamnestic response, but there's no change in these groups. In the nasal wash, you also see that there's very little IgG in the nasal wash prior to infection, and it's much higher in the 30 and especially the 100 microgram dose group. And again, you can see in the nasal wash that again, these responses, there is an anamnestic response in this group. You can notice in the controls that at day 14, they do induce a primary response. Finally, for correlations, we correlated the binding titer to 1351 with the sgRNA in bowel nasal wash or the neutralizing titers. You can see there's a very highly significant correlation between binding titers and reduction in viral reduction of sgRNA, both in the bowel as well as in the nasal swabs. But this clearly shows that, you know, we've done kind of some sophisticated statistical analysis, but clearly shows that increasing the binding and the neutralizing titers would have a dramatic effect on reducing viral replication. And so to conclude, mRNA is highly protective against B.1.351 in the lower airway following high dose challenge. The reduction in viral replication in the upper airway is more limited and only observed in the high dose group receiving 2 immunizations. There was a dose-dependent increase in NEUTs against 1351. The single dose induced undetectable NEUTs against 1351, yet 4 of the 8 animals had a reduction in the lower airway and limited pathology. And so we believe, and this is consistent with many other studies, that there might be antibody-mediated protection in the lower airway in the absence of detectable neutralization. And a lot of work done by Galit Alter, and there's some nice work by Mike Diamond to suggest that antibodies can bind and there might be multiple effector functions mediated by FC. And I also showed you that I think that over several days, there might be anamnestic responses that could quickly respond and account for this. So the bottom take-home message is the current 2-dose regimen is optimal for protection against the B.1.351 variant. The caveat is the limited airway, upper airway protection may in fact be due to the high challenge dose in the model. So here's a study that came out. We had completed our study, but with COVID, everything comes out, you know, on a minute-to-minute basis. And this is a beautiful study published in the New England Journal by BioNTech with their RNA vaccine. And it really highlights some really important points. It shows infection following vaccination against B.1.1.7 and 1351 using PCR. And you can see that with 1 dose against 1351, there's only 17% protection against 1351, but there's 75% after the second dose. And so I think this matches our data quite nice. In terms of disease, you can see against severe disease against 1351, there was 100% protection against severe disease after 2 doses, but nothing after a single dose. So I think it tells us that these primate models might in fact be useful for generating data that might inform clinical development. And I think this highlights why 2 doses are important. The final thing is a little bit of a teaser is we've just completed this study, is we've given 2 shots of 1273 8 months ago. And then they were boosted 8 months later with either 1273 or the matched South African mRNA-1273-351. And the question we were asking is if all of us need a boost in the fall or the winter, would we just get the same vaccine or would we need the variant RNA? There's also studies going on combining these 2, and combining these 2 looks great. And so we boosted them and then we had immune data and we challenged them. I will only tell you that when you look over 6 months prior to the boost, there's no detectable neutralizing titers against 351, and the titers against D614 have gone down dramatically. 2 weeks following the boost, the neutralizing titers go up 10 to 100-fold against D614G and against 351, and they're significantly higher than what you would get after the initial prime boost series. So what I'm telling you is that when we get a boost, it's going to be rather dramatic by waiting this time in terms of neutralization. We've also challenged these animals and we have that result, and it looks quite promising. So let me just close to acknowledge, I have to pay homage, as I always do, to Barney Graham. He's worked his whole life on this. He was way ahead of the game based on structure-based vaccine design and in studying SARS and MERS with his outstanding, at that time, postdoc, but Kizzy's now taken a great job at Harvard to really be ready for this pandemic. And Barney and Kizzy were ready to go with the S2P design at the get-go. And so my hat's off to Barney and his whole crew. I've been fortunate to work very closely with Kizzy, who's just incredible, and well as their team, Annie, Gabriella, and Rebecca. In terms of my own lab, Joe Francique and Barbara Flynn were critical for this. We obtained convalescent samples from the clinical trials program. Nancy's lab is fantastic for measuring avidity. Danny and Matt worked out the subgenomic RNA for measurements. Ian Moore does all our pathology. Adrian leads the team to measure our pseudoneuts or our binding titers with Britta and Sarah. Kathy and Mario Roeder's lab does all the T-cell analysis with her crew. Moderna, we have very close collaboration with Andrea Carpi, Darren Edwards, and Kai Wu, amongst others. Mark Lewis, Hannah, Anthony, Alan, and Laurent are heroes in this whole for not only this vaccine, but many other vaccines. And then these are other collaborators from earlier time points. So at this point, I will stop and take questions.
Thank you so much for that wonderful presentation. We have several questions coming in. So the first relates to whether you saw any IgA measured in the upper airway, and related to that, what you think of the second generation intranasal vaccine candidates.
So yes, we measure IgG and IgA. There is IgA in both the lower and upper airway. It's always much less than IgG, and we only detected IgA in the upper airway with the 100 microgram dose group × 2. So there is detectable IgA, but it's low. I'm a big fan of local getting vaccines to get you antibodies or T cells to sites. So we had done a study with giving adenovirus by an aerosol route, but I am excited and encouraging people to do local immunization in the upper airway.
Okay. So a question on how the contagiousness of the viral variants is measured and quantified, and by what methodology?
No, that's the billion-dollar question. So what you're really asking is, and I think I've hopefully made this clear, that I think all vaccines are certainly having an effect on severe and lower airway disease. And I don't think you need necessarily that much antibody. I think the better vaccines, really the best vaccines, which I think are the RNA vaccines, 2 shots, are your best chance for having upper airway protection and transmission effects. But what I showed you today, that it might be more difficult with the variants than it is against the Washington or the B.1.1.7 strain. And that's consistent with the Novavax data, the J&J data, and even to some extent, the Pfizer data. So the more robust vaccines probably require that for upper airway and transmission effects, you probably require higher antibodies and potentially neutralization. And then the question becomes, is how durable is that? Everything we measure is short term. And so I kind of gave you that teaser at the end that I think once we all get boosted, those responses are really so striking. Transmission.
Right. So can you conceive of a rapid assay platform for screening viral variant strains when they're first identified for immune escape, virulence, and transmissibility without having to do large scale epidemiological studies?
You know, that's beyond my pay grade. I can tell you that I sit on calls with SAVE every week with a variety of really top class investigators around the world, you know, Richard Webby, Betty Korber. They are on a minute-to-minute basis screening and doing epi and doing sequencing. I can tell you that I think we have 3 calls a week, and on every call there's a new variant. And you know, it was 1617.1 first, now it's 1617.2, then there's 1618 from Bangladesh. So every day it's a new variant. The way we prioritize them in terms of vaccine is there's 2 ways to look at it. What is spreading? So B.1.1.7 spreads, but I showed you that it actually doesn't have much resistance and neutralization. So I think our current vaccines are fine for that. I look for the variants that have a massive reduction in neutralization. So like South African or potentially one of the Indian variants, and that's what we choose to use in monkeys, is focusing on that, is what has the greatest cost to vaccine neutralization.
Right, right. So a question about if the local upper airway infection by the B.1.351, is it not producing significant immune stimulation, or are central memory cells less able to localize to the upper airway?
We haven't done enough analysis on T cells in the nose. And that's maybe a sampling issue. So we can detect T cells in the nose, but we haven't done it extensively. The only data that says there's a role for T cells in the nose was a nice experiment Dan Baruch did in monkeys. Is he infected monkeys and then he waited, I think, 8 to 12 weeks when antibody titers were gone and re-challenged them and depleted CD8s. And remarkably, and I think it was remarkable to him, there was no effect of CD8 depletion on lower airway protection. And maybe that's not so remarkable in what I'm telling you, that I think you need antibodies, you have anamnestic responses, but he found that the upper airway, there was a reduction when you depleted CD8s. And that was the natural infection. So it suggested perhaps that, you know, could T cells in the nose be mediating an effect? Remember though, when we give vaccines by IM routes, not aerosol routes, there's probably not that many T cells that are getting there.
Okay.
Okay.
So do you think then that perhaps a nasal boost would be more effective to protect against transmission than an intramuscular after the initial series of vaccinations?
It could. I mean, I have, you know, it's possible that a nasal boost, but what I'm telling you is that if we really do get say a 10 to 50-fold increase in antibodies post-boost, those antibodies are going to find their way to the upper airway. And so I think that it's possible and likely that post-boost, just the antibodies might be able to mediate that role. But, you know, I think it'd be fun to model that question.
And a question about what the clinical symptoms are after challenging the monkeys that were either immunized once or twice.
Yeah, I mean, I must say that the clinical symptoms in the primates are really modest. It's hard to measure really much change in the way of any dramatic changes in temperature or anything like that. As you can see, it's a modest and mild infection. Usually by day 7 or 14, even the controls clear it. So there's not a lot of clinical symptomatology that we get from the primates.
Okay. And are there any data on the protection against the South African variant when someone has natural infection and then receives 1 dose of vaccine?
Not that I'm aware of. That's a good question. I think the questioner probably is aware that when people are infected and get the data on a single boost with RNA, and I think this is you know, been reported by, you know, Florian Kramer and others in Seattle, is fantastic. Is when you get a single RNA boost, your titers go up probably close to when you've had 2 shots or maybe even higher. And so that immunity is quite robust.
Right. And one question here, what are the titers of antibodies in the bowel and nasal secretions?
Well, I didn't want to get into the correlate talk. So we have a way of making of defining titers based on the WHO standard, that we define them in international units. We haven't done that for bowel and nasal swabs yet. We do have those titers in blood where we try to use that metric to correlate with protection. What I will tell the questioner that there's a tight correlate between what we're measuring in blood and what's in bowel and nasal swabs. So when we come up with a blood correlate, at least in our prior models, that will probably likely correlate with the upper and lower airway IgG.
Okay. Can you say anything about the persistence of the responses and guess at whether regular boosters will be required?
Yeah, so I mentioned that at the end. So if you wait 6 months, and I think my colleague Nicole Doria Rose has also done a nice analysis on this from the human samples with Moderna, is that the titers to 1351 are very low to undetectable after 6 months, but they're still detectable against D614G, but they're reduced compared to the peak. So over time, you have probably very low to undetectable responses, at least against 1351. When you boost, those responses exceed substantially what you would have gotten 2 weeks after your initial series. So that waiting that time gives you there's a dramatic effect on the neutralizing responses. So I will have to see how this goes. I wouldn't be surprised in the fall or the winter that everyone might get a boost. I'm trying to sneak away to travel and get out of here in somewhere exotic, and I think I might boost myself.
I think it sounds like a good idea.
Yeah, now bear in mind, the question is when you get boosted, might there be a little bit of reactogenicity, but I can tell you that the data looks great, both in mice, in monkeys, and in humans.
Okay.
There's very nice data on boosting.
Well, we have a lot of curiosity about whether you would be able to hypothesize on a neutralization titer that can protect upon challenge with the different variants. We're asking you to read the crystal ball.
Yeah, all right. So I didn't want to show the slide. It's here. I was waiting for the question. All right, so let me share this.
Okay. Go for it.
Okay. So we've completed an analysis where we gave mRNA from 30 micrograms down to 0.3 micrograms. And then we challenged them with a Washington strain and did a correlates analysis. So this is published on bioarchives, but we came up using the WHO international units that no animal that had greater than 336 international units had bowel sgRNA greater than 10 to the 4th, which we think is 2 logs less than the control and probably protective. And then you can see it took more antibody to protect you against nasal swabs in the upper airway. So this goes along with what I've told you, that more antibody is required for upper than lower airway. So these are 2 metrics. But then I told you we had these dose groups that we dosed down to 1 or 3 micrograms of mRNA. They had titers of 80 to 200 international units respectively, or neutralizing titers of only 50. And they had a 2 log reduction in the bowel. And so we had evidence that that these animals did not protect in the upper airway. So I can tell you for lower airway, it looks like, probably binding titers of maybe between 1 and 300 and neutralizing titers that are either undetectable or very low can protect you short-term against lower airway infection. For upper airway infection, you're going to need more antibodies and potentially neutralizing antibodies. So that, I think, is the simplest way I can answer that.
Okay. And could you comment on the role of T cell responses in contributing to protection, especially in the context of suboptimum antibody levels?
Yeah, so as a T cell biologist, there's actually wonderful work by Shane Crotty and Alex Sete and others showing that there's beautiful T cell responses induced by primary infection. They're conserved against variants. And I have no issue that over time, as antibodies go down, the T cells could have some effect in the lower airway against disease. There's very little experimental evidence in monkeys to prove that. And that's actually a tough experiment because you'd have to give a vaccine, wait a long time till titers got really low, and then challenge them and deplete them of T cells and look to see if there's an effect. So I have no data to suggest a role for T cells. What I can tell you is antibodies, we've just written a paper on correlates. You can passively transfer antibodies to hamsters, they mediate protection. So it's a mechanistic correlate. So that's direct evidence that they're sufficient. So I think that antibodies are the first line of defense, they're critical. And I think that there could be a role of T cells later on. And it would depend on the vaccine. As you know, the protein vaccines don't induce CD8s. So the RNA and the viral vaccines do. And so perhaps they have a role.
Right. And just as a promotion, Alex Sete will be doing a talk at the Global Lab meeting in June. So
I wouldn't miss that.
For that.
That'll be a great talk. And Alex can give you all of the data to support a role for T cells.
Right. Okay. So we have a question on whether the mechanism of protection by dose is based on the broadening of the immune response.
Yes. So in the correlates paper, dose gives you obviously better immune responses, but in the correlates paper, by doing dosing down, we actually show that antibody is the correlate independent of dose. And so, no, I'm not sure necessarily that would be the broadening response. It's a very good question. And we, you know, as far as we can tell, dose did not affect avidity. So there's no change in dose with avidity. And so unless there's something more cryptic where we would do mapping. So Nancy Sullivan's been doing some mapping. So that's actually an interesting idea. Maybe in fact, there might be more breadth with higher dose.
Right. We had a device question. Do you have a standard device in order to take swabs to have a uniform volume for antibody quantification?
Biocall does it. I don't know what device they use. We take a standard amount, and Matt Gagnon and Danny Dewick's lab have spent a lot of time optimizing how you collect it to make sure that you don't degrade the RNA. And so it's in the methods of our paper, but I encourage you to get that because he showed that most standard ways of doing it were grading 90% of the RNA. So he has a way to preserve it. So we get, kind of better data. That's might be another reason why we're seeing less protection in the upper airway is that our techniques have improved for quantitation.
Right. And we had a question that there was a change in the viral concentration during the challenge study. Did it have any effect on the result?
No, the viral concentration we used for the challenge was 5 × 10⁵ PFU.
Okay.
And we had based on our pilot study. That is based on a quantitation done by Mehul Sutar on virotempris cells. It actually could be 10-fold higher if you based the dose on quantitation from virotempris ACE2. So this is for the aficionados in the crowd who would say, how do you base your stock concentration? So I think we're being conservative. It's possible that dose was actually 10-fold higher. And I think it's frankly too much. I mean, we're getting 10 to the 6th to 10 to the 7th logs. There's not a lot of data published in humans using subgenomic RNA in nasal swabs. There's one from back about a year ago, and our values are at least meeting that threshold, if not significantly higher. So frankly, I think it might be better to lower the dose a bit.
Okay. And I know you've said that there isn't so much data available on the T cell responses, but we do have a question on whether there's any information that the J&J vaccine might elicit better CD8 responses.
Well, yeah, I mean, you've seen the human data. RNA vaccines do induce CD8 responses, and the J&J vaccine and the AstraZeneca also induce CD8 responses. So I certainly wouldn't preclude a role for T cells potentially with the J&J vaccine. and I think, it'll just have to come out. But yeah, they induce good T cells.
Okay. So what's next after your trip to your exotic locale?
Well, we have a cool malaria finding. We can
Oh.
prevent malaria with an antibody short term. So I'm hoping to get back to my day job.
Right,
Back to malaria and TB. I think the variants have thrown us for a loop. But just as an optimistic message, I think all our vaccines, there's certainly these RNA vaccines and others, and the protein vaccines all look very good for protecting us against severe disease and lower airway disease. Whether there's a nuance with upper airways, we'll see. RNA is great because it's flexible. They've already made an RNA for 351. We're going to be testing an RNA for the Indian variant. So the turnaround is really quick. And so I think if we all get boosted, and frankly, we need to get the vaccines in the rest of the world. It's just an amoral that we can't get vaccines to the rest of the world. And so we'll all be fine, but the world needs to be fine. And so I think the science is good. It's just a distribution issue.
Yeah, absolutely.
All
Well,
right.
thank you so much for that fantastic presentation. I really appreciate you taking the time to share your work with us and for answering the questions. Thank you so much.
Sure.
I'd also like to thank all of the attendees for participating in today's webinar. We are fortunate to have such an engaged audience for these lab meetings, and I thank you for joining us and for submitting such great questions today. I also want to invite you to join us 2 weeks from today on June 3rd for the next Global COVID Lab meeting. Our speaker that day will be Dr. Douglas Nixon from Weill Cornell Medicine. His presentation will focus on COVID-19. Can old vaccines have new tricks? And if you're interested in more research on COVID-19, please sign up for the HVP COVID Report, a bi-weekly newsletter that provides insights from experts around the globe and highlights the latest scientific articles and data. Finally, please visit our website and follow us on LinkedIn, where we will upload a recording of today's webinar. And with that, I'll say thank you again for participating today. Stay safe, and we hope that you'll join us again on the next Global COVID Lab meeting. Thank you so much.